Okay, good afternoon, folks. How are you doing today?
Good.
How you doing?
Hello.
Okay, hope you're staying warm. We're getting some chilly days today, this week. Okay, so, all right, folks. So, I was looking at the syllabus here, just taking a look at our schedule, and we're moving right along, as they say. Basically, we're basically 3.5 weeks away from your third lecture exam. Third lecture exam will be the day you return from Thanksgiving break. Okay, so things are really accelerating here. Okay, we have a lot to do, but we can get it done. So, do you happen to have any questions about our schedule at this moment? Okay, all right. Well, let me know if you do. All right. And so today we're going to start class today by reviewing the workbook assignment, that assignment number 3 on page 8 of the workbook, and then doing an in-class activity together in breakout rooms. So, as I've mentioned previously, for lecture exam 3, you will see an increasing emphasis on lecture exam 3 of questions that require an interpretation of scientific data that's presented to you in a graphics format. Okay, so that's why we're doing the workbook assignments, to get you up to speed with some basic ideas about how to interpret information that's presented graphically. Okay, and so we'll get started with that. Just waiting on one more student to get logged in here. Okay, so let's get started. If you would please, have your workbook out at this moment. We're going to go through the questions that are presented in the workbook, give each of you a chance to contribute some answers here and have a discussion about this. Okay.
Excuse me, Professor, I just want to let you know I'm driving. I'm almost home, so I don't have my book in front of me right now.
Okay, Tiffany, please focus on the driving. I don't want you to get in an accident. Okay. All right, so I'm going to share my screen with you. And okay, so there we go. Okay, so we have workbook assignment number 3. Gang, it is totally okay if you didn't get answers for everything. It's okay, but we want to be sure by the time we're done today that you truly understand the answers to these questions and you're not feeling uncomfortable. Okay, so please speak up along the way if something is bothering you. Okay, so here we have some scientific data that's been presented to us in a scatter plot. A scatter plot is an XY plot with these little dots here, right? And lines have been drawn between the dots through a process which you don't have to worry about, but it's called interpolation. Okay, and so we're going to see how you did with this. The data in this is actually from a real scientific paper. Thank you, Rachel, appreciate it. And so I didn't make any of this up. It's not like Dr. Allen is going off the deep end and just throwing random numbers out there. All the things in this workbook that I'm throwing your way come from legitimate scientific publications. Okay, so let's start with the first question. Okay, the first question is, which axis is used to plot the independent variable? So I'm just going to ask you to please contribute your response to question 1 in the chat window. Okay, and you can go ahead and just take a look at your peers' answers, and it seems like there's quite a bit of consensus here, right? I don't think there's any arguments here with any other answers. So you're either all right or you're all wrong. And folks, you are absolutely all correct. Yes, the answer for question 1 is the x-axis. Okay, and so, I'm not going to type these things out in any official way, but, since you've been doing the work, if you do want to make some comments on your workbook, if you've got something wrong, you need to correct it, please do that. Okay, so the x-axis. All right. So let me ask another question here. In relationship to question 1. So here's my next question and coming in here, and this is not in your workbook, so this is just an extra question I'm throwing at you at this moment. Okay. Here it comes. Is the x-axis used to plot the independent variable because there's something special about that data, or is it done this way due to historical convention? Okay, what do you think, folks?
Is it the historical convention?
Okay, thanks, Tiffany, for participating there. Okay, yeah, absolutely. Okay, I'm seeing a lot of good answers in the chat window this morning. That's very encouraging. It is absolutely historical convention. There's nothing special about the x-axis data. There's no reason we couldn't put the x-axis values on the y-axis and vice versa, okay? So it's, historical convention is correct. Here's my next question. Here it comes. If I chose to plot the x-axis data on the y-axis and the y-axis data on the x-axis, would the relationship between the variables change? Okay, good, folks. You're absolutely right. The relationship would not change at all, would it? It would not change. Here's my next question coming into the chat window. Okay, you're doing fantastic so far. Okay, here's the next one. Here we go. If I chose to plot the x-axis data on the y-axis and the y-axis data on the x-axis, would the graphical presentation of their relationship look different? Okay, I'm very encouraged by your responses today. You're absolutely right. You're absolutely right. Just for giggles, gang, I'm going to do something with you here on the screen, okay? So, all right, so hold on a second here. All right, so I hope you can see the screen okay. Let me boost this up a little bit. All right, so this is a piece of software I use in my in areas of my life where I need a bit more technical information. And at any rate, so let's say I produce some random data here, okay? And so I'm making a bunch of XY coordinates, right? I have -10 and 100, -9 and 81, so on and so forth. So basically I'm going from -10 with the X value up to +10, okay? So let's say I do that, okay? And then I plot that data. Okay, here's what the data looks like when I plot it. Okay, you can see for yourself, it basically looks like a U. It basically looks like a U. Now, folks, if I take the data that I've created here, okay, if I take this data that I've created here, and I take each XY coordinate and I switch the position of the X and Y values and get a list like this, where now 100, which was the Y coordinate on the first list, is now the X coordinate on the second list. So I'm switching each of the X, Y coordinates of the first list. I'm switching their position in the second list. Okay, so we've already established in the chat window that the relationship between those values is not going to change. However, here's my next question in the chat window. Can you predict how the graph of the reverse data is going to look when I plot it for you? When I plot the second data set, can you predict? Okay, so I have to be careful with my questions because I realize what I intend and what you interpret is going to be different. Okay, yes, you can predict. So let me ask you, what's it going to look like? What's it going to look like? Okay, all right. So, all right. So I am definitely seeing some correct answers. I'm also seeing some incorrect answers there. Okay, so let's take a look at this and see what it shows up as. Okay, so remember, the first graph is what you see here from the first data set. Now I'm going to plot the second graph. Here we go. 1, 2, 3, bang. Okay, so for those of you that said it's going to look like the letter C, you are spot on. Good for you. Good for you. Okay. All right. So am I going to ask you to do that kind of thing on an exam? No, I won't. I promise I won't. But I just wanted to throw it out there for those that maybe haven't spent too much time thinking about this kind of thing. Okay. You're doing very well. Keep going. Okay. So let's keep going, folks. Let's come back to the workbook assignment. Okay, so question number two, please put your answer for question 2 in the chat window. What did you put for question 2? Define the independent variable. Okay, so, and as everybody's putting their answers in, just go ahead and read across some of them and see if they mean the same thing as what yours is, right? Compare other folks' definitions to yours. Let's just read through a couple of these. An independent variable is a variable that is not affected by another variable. Okay, the variable causing change on the other variable, very good. The variable that is the same for each sample. Okay, the variable that changes. Okay, so Dallas, that's a little bit of a weak definition. Definitely look at some of your classmates there. Okay, the independent variable is the thing that is getting changed or manipulated. Aubriana, I really like your definition. Okay, Elena, who is niece, the variable that can be manipulated or changed, absolutely. Victoria, it's not affected by anything else. That could be a bit of a dangerous statement, Victoria, but we can run with it. Yasmin, independent is something that is manipulated or changed. Okay, a variable that cannot be affected. Brianna, be careful with that one. I think you're being a little bit too rigid on there. And Morgan, the variable that causes change of effect on the dependent. Taylor, the variable that is controlled by whoever is conducting the test. Okay, I like that too. All right. Thank you, everybody, for your contribution. I generally agree with most of the definitions. Okay, the independent variable is going to be the variable that the human being, the student in school, the scientist in the lab, whoever we're talking about, mom and dad at home doing an experiment, school experiment with their kids, the independent variable is going to be the variable that is manipulated and adjusted by a human being. Okay, so question 2. All right, independent variable is controlled and manipulated by a human. Okay, so question number 3 there asked you to circle the label on the x-axis. Obviously, I can't get you to show, I can't really get you to show what you labeled. I mean, I suppose I could ask you to share your screen, but for right now, for question 3, please type in the title that you circled in the chat window. For question 3, what did you circle? Type in the words. Okay. All right. Very good, folks. You're absolutely right. The x-axis is, from our typical viewing perspective, the horizontal one, right? So that means the label would have been concentration of antibacterial drug named APP in milligrams per liter. Okay, very good. Let's keep going here. So let's see here. Question number 4. Question number 4 is going to be very hard for me to get you to respond in some way. So tell you what, I'll give you the answer to question number 4 by drawing it on the screen in front of us here so we can see it, okay? Question number 4, draw an asterisk on the graph above for the following coordinate, 2000 megs per liter at minus 20%. Okay, so I'm going to draw an asterisk on the screen at that coordinate. And that coordinate would be roughly here. Okay, roughly here. Hopefully you can see that okay. So let me ask you in the chat window, and please just give me an honest answer because we're all friends here and nobody's getting graded on this, okay? Does your asterisk match the position of my asterisk? Okay. Okay, is there anybody that needs help with this? Okay. The reason I'm asking is because sometimes students forget that the 0 line is here, right? The 0 line is not in the bottom right corner, right? So on the x-axis, you do have 0 here, but on the y-axis, 0 is actually in the middle of the graph. So sometimes that throws students off. So my word of caution for you, particularly on exams, is make sure that you know where the 0, 0 coordinate is in the graph, okay? Just be careful about that. Okay, so if yours matched right there, you're doing great. Okay, let's continue. Question 5, please give me your prokaryote for question 5. Okay, all right, looks like we're getting some good answers here. Okay, there were 3 different alternative correct answers here, right? There's 3 options you could have put down for this one because there are in fact 3 bacteria plotted on this graph. And so it looks like you're all doing fine, right? So Staphylococcus aureus is a correct answer, Bacillus cereus is a correct answer, and Escherichia coli is a correct answer. Okay, so obviously the only remaining answer for the eukaryote is going to be the Saccharomyces cerevisiae, which I won't bother to type in grotesque detail, but the eukaryote would be Saccharomyces cerevisiae. Please let me know if you have any questions. Question 6. Would you folks put down for question 6? Which organism experienced a 50% growth reduction at 1,000 mg per liter of APP? Okay, all right, gang, you're doing great. Very proud of you. You're doing fantastic. Bacillus cereus is the correct answer. Good job. please let me know if there's something you don't understand. And what I mean is, what I'm worried about is that there may be some students sitting at home saying, oh, I didn't get that, but I'm afraid to speak up. I don't want you to do that. If you are confused by something, reach out to me. Please come see me during office hours. Okay. Question 7. Question 7, from this data, is APP better at inhibiting the growth of a gram-positive organism or the gram-negative organism? Okay, looks like everybody's saying gram-positive. Seems to be the consensus, and I wholeheartedly agree with you. You're absolutely right. If you look at the data, right, the blue line here gets highest on the graph, and higher on the graph represents the best growth reduction. So that's a gram-positive organism. So you have the right answer. Okay. Question 8. Question 8. Which concentration of APP reduced the growth of E. coli by 65%? What did you put down for question 8? Okay, so all right, so let's take a look now. Okay, now take a look at your answers. Okay, now take a look at your answers. There is some disagreement, right? There's some disagreement. Okay, all right, so I see, so Taylor's changing his answer. Okay, very good. So right, 2000 would definitely be a little bit on the low side, wouldn't it? Okay, so folks, on an exam, when I'm asking you for something like this, where you might have to read values off a graph, I will give you a little bit of flexibility. I mean, as long as you're within the correct ballpark, I'm going to give you credit for it. But I would say that 2000 is a little bit too far out of the ballpark, right? And so let's just be sure we're okay, right? E. coli is the green line. The green line. So we're looking where the green line goes past 65%. So 65% is going to be roughly up here. I'm just very roughly tracing it across. And so we're just about at 3,000, just about at 3,000. So if your answer is in the neighborhood of 3,000, when I say neighborhood of 3,000, I'm saying 2,800, 3,200, you can expect that I would give you credit on an exam. Okay. All right. So be very careful about reading information off a graphic like this. We want to be sure that we don't suffer from overly crooked eyeball syndrome, right? We want to be careful we're really looking at it faithfully. Okay, any questions or problems about that? You know, looking at something wrong is a hazard for every one of us. It's just that there are certain circumstances in life where looking at something wrong and then doing something because you got wrong information could really have a serious consequence, right? I mean, it's not just a point off on an exam. It could be a patient that got too little or too much medication. Think about that. Okay. All right, let's continue. Question 9. Question 9. At what concentration of APP does Staphylococcus aureus and Bacillus cereus exhibit no growth at all? What did you put there? Okay. All right. Okay. So yeah, you're going to be very high. You're going to be very high on the graph and over on the right side, right? So we're looking at things that start where the slope of the curve starts to become zero, where you fully get kind of a perfectly horizontal blue line up here. So for those of you that are saying 3,800 to 4,000, I agree with you. 3,500 is a little bit of a stretch for me to get to. But it's not a horrible answer, Samantha. Alicia, 2,000 is definitely not good there. Alicia, do you understand why the answer is 4,000? Do you need me to explain any of that?
No, I understand.
Okay, all right. And if you need to come chat with me, that's quite okay. Feel free to do that. All right, so number 9 is somewhere around 3,800 to 4,000 mgs per liter. Okay, next question. Question 10. Question 10 asks, which organism or organisms had their growth stimulated rather than inhibited by APP? What do you say, folks? Okay. All right. Okay, it's kind of interesting to take a look here, right? I mean, it's helpful if you can see how your classmates are answering too, right? It gives you confidence that everything's okay, or maybe something's not okay. And so the answer, the only answer really is Saccharomyces cerevisiae, right? The orangish red line down here at the bottom, right? Okay. So maybe it strikes you that this drug seems to be stimulating the growth of one microorganism, yeast in this case, and it seems to be reducing the growth of the other microorganisms. So let me ask you this question in the chat window. What could explain this difference in stimulation versus reduction of yeast versus bacteria? What could explain this? And please understand, I'm not telling you as the instructor that I know the answer. Okay, Taylor, I like your response there. You're saying APP seems to be an antibacterial drug, not an antifungal. I think that's a very reasonable response. Okay, and so, okay, so Rachel, remember that S. cerevisiae is not bacteria, it's yeast, which is a fungus, right? So S. cerevisiae is not a bacterium. Okay, hold on a second here. So getting back to this conversation here, I think the most realistic thing we can say explaining this difference in response to the drug is that the yeast, which is S. cerevisiae, is eukaryotic, right? It's eukaryotic. And the other 3 cells are prokaryotic. Okay, there is something truly significantly different in the metabolism of a eukaryotic cell, in this case, the yeast cell, versus the metabolism of the prokaryotic bacteria that's triggering this difference. You can see for yourself, Afala Shadi, I see your question, just hold on. You can see for yourself that at relatively low dosage, low concentrations of APP, there is a significant stimulation of growth of S. cerevisiae. Okay, so all right, before I come back to Falashadi's question, folks, do you have any questions about the answer to number 10? The fact that Saccharomyces cerevisiae actually had its growth stimulated, not reduced. Okay, so let me come back in the chat window for Afala Shadi. For question number 9, did you mean 4,000 for both? I meant 2,000 for Staph aureus and 4,000 for Bacillus cereus, I guess. Okay, so Falashati, if we take a look at the graph there, the Staph aureus is the blue line, right? And the Bacillus cereus is the golden yellow line. And so the golden yellow line is still showing a slope there, right? It's still a very gentle upward curve there. So that means its growth, it is still growing a little bit there, right? We would need to get to an absolute horizontal position to show no change in the growth. So that doesn't really get horizontal at all. Probably at most approximates horizontal up there when it merges with the blue line. So Falashati, I meant 4,000 for both Staphylococcus aureus and Bacillus cereus. Okay. Are you okay with that, Falashati? Okay, I see your response. Okay. All right. Okay. So gang, let me ask you a question in the chat window here, and I'm coming back. I'll say, so question 10, the answer to question 10 is is Saccharomyces cerevisiae, the yeast. That's the answer to 10, S. cerevisiae. And so let me ask you in the chat window, what did you put down for question 11? Question 11 says, what does it mean that Saccharomyces cerevisiae has data plotted as negative percentages? What does that mean? Okay. Thank you, Taylor. How about the rest of you folks? Okay. All right, good. I think you're getting it. Fantastic. That growth was actually stimulated. So if this kind of logic of, no, wait a second, what are you talking about? There's a negative value and you're telling me that there's a stimulation of growth? That seems stupid. I can understand why you might be uncomfortable with that. So let me throw this logic at you in the chat window. Here we go. What is the product, meaning multiplication, of -2 times -3? Okay, folks, you got it. Good, it's +6, right? So what we're saying here is that we have a negative value on the y-axis, and the label of the y-axis is saying everything on the y-axis represents a reduction in growth. So if we have a positive y value above the y = 0 line, that truly represents a reduction in growth. But if we have a negative value, it's like multiplying two negative numbers together. -2 times -3 is a +6, positive being a stimulatory effect, okay? So let me ask you, are you okay with this? Okay, it's kind of a non-traditional way of presenting information, isn't it? I mean, it's not something that you might pick up or can think of right away. Okay. So
Doctor, I want to ask you a question in the chat room.
yeah, absolutely. Oh, okay. Thank you, Elena. Let me come back to that. So Elena, here's your question. Can it be interpreted as antibiotic resistance? What do you mean by it, Elena?
The Saccharomyces cerevisiae.
Okay, so
That antibiotic is not effective on it because it's growing.
okay, thanks for asking that, and let's pick that apart a little bit. Okay, so we're looking at the orangish red line here. So first off, I would say this. So Elena, I'm going to speak to you right now, but everybody should be thinking about this. So Elena, is the response of this yeast cell the same at every concentration of the drug?
No.
Okay, beautifully stated. So let me ask this, Elena. Do we get more stimulation of the yeast growth on the left side of the graph or the right side of the graph?
On the right side. Wait, no, wait a minute.
As you're thinking
I
about
understand
it,
the question.
that's okay. As you're thinking about it, let me just restate the question for everybody. So look at the yeast line there, the orangish red line. Is there more stimulation of yeast growth on the left side of the graph or the right side of the graph? What do you say, Elena?
It's on the left because on the right, it's the reduction is growing. So on the left, it's still growing. It's not being reduced.
Okay. We get muddled around with words, don't we? This is a very kind of convoluted way of looking at things. So let's pick this apart. So on the left side of the graph, we have the largest negative numbers, right? Down here, we're at like, we're at like -90, -80, -70. So we have a very strong growth stimulation at this point, Elena. And so I would say to you that at lower concentrations of this drug, not only is this yeast resistant to something in the antibiotic, but it's actually being stimulated. It actually enjoys the drug. It actually likes the drug because it's promoting growth. And then as the concentration of the drug increases, the metabolism of the yeast cell starts to choke on the excess amount of drug so that the negative values start to become more positive, which means we are reducing the stimulatory effect and starting to increase the inhibitory or reduction effect of the drug. So is this cell antibiotic resistant? Well, first off, that phrase doesn't mean too much in this conversation, right? Because this drug, antibiotic, means antibacterial. It does not mean antifungal, okay? But we could say at the lower doses of this medication, the drug is stimulatory for growth, and at higher doses of the medication, it is it's almost a wash. It almost does nothing. Okay. All right. Did I make it better or worse for you, Elena? All right. Honest answer. Very good. Okay. So yeah. I guess I would say to you at this point.
I get it. When you broke it down, it just takes me a bit to kind of think about this stuff. I'm not very good with numbers.
It's okay. It's okay. And this is kind of an awkward way of presenting information. And honestly, I did this to all of you kind of intentionally, right? I wanted you to look at this, okay? And so once again, we have some data here which shows that a drug, at least at relatively low concentrations, is actually stimulating the growth of a eukaryotic cell. And normally this drug would actually inhibit the growth of prokaryotic cells. Again, highlighting the fact that there's truly something different in the physiology of bacteria versus yeast, or bacteria versus human cells, okay? So another question that comes up here is, why in the world would anybody want to show growth reduction as a positive value? Why in the world would any scientist want to show the reduction of growth as a positive value? Let's take a look at this, folks. I'm going to throw something your way. Okay. So again, I hope you can see this all right. And this information I'm throwing at you is in relation to my last question. Why in the world would any reasonable person want to show a reduction in growth as a positive number? Why would you do that? That seems very odd. Okay, so let me do this. So imagine for a moment that we compared the growth of a microbe without any drug present, to the growth of the same microbe in the presence of either drug A or drug B. And let's say that we get these results. Okay, so here you have a graph. I'm showing you the amount of growth of a microorganism where there's no drug present, and you can see it gets up to a value of 1. That value of 1 is arbitrary. Maybe it means that we have a bacterium on a petri dish, and without the presence of the drug, the microbe created a colony that had a diameter of 1 inch. So 1 inch could mean anything. It's simply a number, okay? So this is how much the microbe grew when there was no drug present. Drug A shows you how much the microbe grew when drug A was present, and drug B helps show you how much the microorganism grew when drug B was present. So here's my question for you folks. Please type into the chat window at this time, what do you think the answer is to my new question number 1? Does drug A or drug B show more change in growth? Okay, I'm just going to wait for folks to come across here. Okay, all right. And so let me ask you this, and so let me state the question again, right? The question is, does drug A or B show more change in growth? And here's the answer to my question. Here we go. The amount of change in growth is the same, but in opposite directions. Drug A does not show more change in growth than drug B. It does not show more change in growth than drug B. It's the same amount of change in opposite directions. So let me work through this with you. The amount of change is the same, but in opposite directions, okay? The growth of the microbe in the presence of drug A is 4 times the growth with no drug. In other words, if you took this no drug bar, you could fit it into the drug A column 4 times, 4 relative to 1. The growth of the microbe in the presence of drug B is 1/4 the growth with no drug. In other words, this drug B bar could fit into the no growth column 4 times. If we stack the drug B bar four times total, it would be the height of the no drug. Okay, so before I go any farther, and you can give me an honest answer. I always want you to give me an honest answer. Are you convinced yet that the amount of change is the same, but in opposite directions? Are you convinced? I mean, if I haven't convinced you, say so. Okay. If I haven't convinced you, say so. Okay. So, all right. And so here's why I'm doing this. Remember, our original question was, why would anybody show a growth reduction as a positive value, right? That was the question. Why would any reasonable person do that? And so here's how I would answer that question. If you got this question wrong about does drug A or drug B show more change in growth, then you now know why researchers often choose to show reduction as the drug A bar instead of the drug B bar. The visual, meaning psychological impact of change is greater with the larger bar, is it not? We look at that larger bar as animals and say, well, that's a bigger difference, but it's not. It is not a bigger difference. We're comparing, in the first case, we're pairing drug A to no drug, and in the second case, we're pairing drug B to no drug. The amount of change is actually the same, but it is in the opposite directions. Okay, so yes, Chelsea, I have bamboozled you. Okay, but I'm trying to point out that as human beings, we can be overly impressed by big things, right? We can. I mean, everybody likes a big house, a big car, a fast car, right? We can be overly impressed with these things. You're right, Elena. Scientists don't think like normal people. We're very abnormal. I agree with you, okay? But take a look at this last sentence here. The visual impact of change is greater with a larger bar and may influence the decision of an investor looking to finance the drug. If you're a drug company and you have a drug that reduces growth, it is better to show the reduction in growth as a positive gigantic bar than a tiny little bar, is it not? Because you see how you yourselves interpreted this. So,
Dr. Allen, I think that
Yes.
scientists think more in numbers differently than what we do, because you deal with very, very large amount of tiny. So
Okay.
your thought process is a lot different. It's like more, it's just different from what we normally are used to.
I agree with you, Elena, absolutely. And you know what? The more you do this kind of thinking that I'm throwing at you right now, the more comfortable you get with it. In other words, I'm trying to get all of you to come over to the dark side of the force, right? That's what I'm trying to get you to do, okay? And so here's the idea that I want to put into your brain. Think about how many times we as just members of society have information and data thrown at us from people at school board meetings, from people, from politicians, right? From Congress, state legislatures, you know, who knows what it is. We are always presented with visual information of various kinds. And the point I'm trying to make is, are you sure you're interpreting the data correctly? Because even though there's nothing false with this bar graph, the bar graph is what it is. Because of our psychological predispositions to interpret things a certain way, maybe we kind of get bamboozled a little bit and come to the wrong conclusion. So scientists are not immune to this. We're not perfect, we screw up. And so let me show you something about how you can take this relationship and make it more interpretable to even the average Joe, okay? I don't mean that in any negative way, because I'm an average Joe too, all right? Very soon in class, you know, remember at the start of the course, I said, I want you to have a calculator, and that calculator's got to have a log button in it. Very soon in the course, we're going to be pressing those log buttons in the calculator. So let's continue here. Consider what happens if we take the base 10 logarithm of each number plotted above. So the numbers above are 4, 1, and 1/4. All right. So if I take and put those numbers into a calculator and ask it to give me the logarithm of them, here's what I'm going to get. I'm going to get 0.60206 for 4. I'm going to get 0 for 1, and I'm going to get negative 0.6026, excuse me, negative 0.60206. Anyway, you can see that the log of 4 is positive 0.6 and the log of 1/4 is negative 0.6. And if I replot that information with the bar chart, okay, folks, let me ask you again, in the new bar chart, which drug created more change? Is it easier for you to get that it's actually the same amount of change in the opposite direction? Okay. Okay, so this is what I'm trying to feed into your brain a little bit to think about this. And while you yourself will not have to be a mathematical whiz, I know you have stress with math, I know that. Okay, but there is growth that comes through stress. That's why we're doing this. You don't have to be a mathematical whiz, but in the future, you'll have to be able to use the log button on your calculator, okay? So what I'm trying to do is present an argument for why logarithms are actually very useful mathematical beasts in science, because they can take changes that kind of seem different and expose the reality that the change is actually the same, but in opposite directions, okay? And once again, it highlights the fact that our primate brains can be very easily tricked with numbers and the relationships between numbers, okay? So let's, any questions? Okay. Folks, I think by and large from the conversation we're having here today that I'm very happy with generally the answers you've given. So, you know, don't worry. I think you're doing quite good right now. And let's just finish this up. Question 10, excuse me, question 12. What'd you put down for question 12? Okay, it is in fact 4 milligrams per milliliter. Do any of you need me to write out the work for this? Do you understand the work for it?
I understand what to do, but can you show us, just because
Okay,
I think I have them maybe flip-flopped a little bit, but I knew what I needed to do.
okay, sure thing. I'll just write it out on the PDF document here. So I won't be sending this to you, so you won't get it from me. So you'll have to write it down if you want to document it. Okay. All right. So okay. So what I'm going to do is take 4,000 milligrams. That's per liter, right? So that's over 1 liter. And I want to convert it to mgs per mil. Well, there's nothing I have to do with the milligrams. So what I'm going to do is create a conversion factor for the liters. And we know that I have 1 liter over 1,000 milliliters, right? That allows me to cross off the liter on the diagonal, cross multiply. That's going to be 4,000 milligrams over 1,000 milliliters. And if I simplify that fraction, it's going to be 4 over 1, or 4 milligrams per milliliter. Okay. All right.
All right, thank you. Because I knew that there was 0.001 milliliters in a liter. So I just multiplied the 0.001 by the 4,000.
Okay, I'm not sure I agree with that first statement you had, but I'll let it rest unless you want me to dig further, Rachel. Maybe that's something we should talk about later,
Or
Rachel.
I had it backwards. It was 0.001 liter per milliliter.
Yes, that
So I
can,
multiplied those.
that I can agree with. Yes, absolutely. Okay, folks, good job. So if there's no further questions on this activity, any questions anybody like to ask? The fact that really basically all of you could tell that the answer to question 11 represented a growth stimulation makes me feel very good. I think that's wonderful that you figured that out. Good job. Okay, so what I'd like to do now is, in our remaining time, get you into some breakout rooms so that you can work on the second workbook activity. Now, I know that some of you had worked on it, but I'm hoping you all did that, because my intention was that you could team up and work on this a little bit together, a little bit of camaraderie, if we will. So I'm going to put you into small breakout rooms and ask that you please talk to each other and try to solve these problems together as a team effort. If you went ahead and did some of these things, then please be part of the conversation, but please don't hand out any answers. I don't want you to hand out answers. I want you to be part of a conversation where your group arrives at an answer by collectively understanding the questions, okay? So I don't know how long this might take. We might need to take the rest of the lecture and then continue it in lab a little bit. That's okay with me, but I simply want to get this process started, okay? So do you have any questions about what I'm asking you to do. I'm gonna ask you to work as a team to answer the questions of workbook assignment number 2 in breakout rooms. Okay, so folks, I think what I'm going to do is make the breakout rooms have 3 or 4 people, 3 or 4 people. So hopefully you can get some comfortable interaction there. Please talk to each other. Please work out the answers to the best of your ability. I'll be staying online with you. If you have a problem, shoot me a message. I'll come into your breakout room, see if I can point you in the right direction, okay? So breakout rooms are open now. Go ahead and join, see what you can do. Hmm.
Oh, sorry. Had to go to the bathroom.
That's okay, Taylor. Okay, folks, let's see how you all did. Let's see how you all did here. Okay, so I'm going to share my screen. Okay. So, let's see. let me, well, I can't really show, well, maybe I can. Let's say it's hard for me to show the graph and write responses at the same time. I don't have a lot of screen real estate, but let's see here what we can do. Okay, so we'll take this down a little bit. Okay, so there we have the graph, and I will try to squeeze some answers here. I'm not going to send you this. I am not going to send this document to you. So please take your notes if you need to for something, okay? All right. So question 1, what units of measurement are used to express enterotoxin B concentration? That would be micrograms per milliliter, right? Which you might have written down symbolically. So you can insert symbols and words. So you might have had this kind of thing, microgram per milliliter. Okay. All right. Question 2, define concentration in language that you understand. Obviously, your language would probably be a bit different from my language, but the basic idea is something like this. So concentration is a ratio of the quantity of material, which we expressed as a mass unit, dissolved in a volume of liquid. There certainly are alternative ways you could define concentration, but this is one way that is pretty standard from a biology perspective. A ratio of the quantity of material dissolved in a volume of liquid, such as micrograms per milliliter, right? All right. Question 3, circle the label used on the dependent variable axis above. Well, you would have circled the label on the y-axis, which says toxin, right? Toxin, micrograms per mil. Okay. Number 4, define a dependent variable. The dependent variable is the variable that the experimenter, the human being doing the experiment, does not intentionally manipulate. Okay, so it's the variable that responds to the independent variable which the scientist is manipulating. So I think probably the simplest way to define a dependent variable is to say it is the response variable. Okay, it is the variable that responds to changes in the independent variable. Okay, question 5. What media pH generally supports the highest level of enterotoxin B secretion over time? And so I hope you came up with a pH of 7. Question 6, is the pH value you answered for question number 5 acidic, basic, or neutral? You know, if it's literally 7.0, that in fact would be a neutral pH. Question 7, is enterotoxin B manufactured inside or outside a cell? Well, cells always make useful things inside their interior and then tend to pump them out of the cell. So the answer is inside. If you know, I don't know that you have any special knowledge of enterotoxins at this point. You will later on in the semester. Enterotoxins are almost always proteins, and you know that proteins are made inside cells. Question 8, is this graph reporting the concentration of enterotoxin B as it exists inside or outside the cell? Well, it's outside the cell, right? It's outside the cell, okay? And you know that because the title of the graph, okay? The title of the graph says the influence of media pH on enterotoxin B secretion during fermentation, right? That word secretion is telling you that the cargo is being put outside of the cell. Okay. Question 9, define the term secretion in language that makes sense to you, that you understand. Secretion is the process of exporting material from the cell. Please appreciate that the term secretion does not imply biological activity. In other words, you know, maybe in a ridiculous analogy, the store Best Buy secretes a brand new TV to your house, right? They export the television from their store location and ship it to your house. It doesn't mean the TV is actually going to work when it gets to your house, right? TV could be broken for some reason. So please understand that secretion does not imply biological activity. Secretion does not imply biological activity. Okay, so there's a couple questions coming up that I am interested in getting your responses in. So folks, if you would please type your answer for question 10 into the chat window. What'd you say for question 10? What is the concentration of enterotoxin B on line A at 7 hours of growth? Okay, all right. I'm quite content and happy with what I'm seeing there. Yeah, you know, if you're in the 210 to 230 ballpark, you know, I had answered 220. I completely agree with you. That's a good answer, right? So don't forget, the question is, what is the concentration? You can't write a number down. You got to write a number and a unit, right? Units matter, folks. Units matter. So micrograms per mil would be part of your answer. About 220 micrograms per mil. So good job, folks, on that. Question 11. Question 11 says, how many additional hours of growth does it take line C to display a secretion value that is greater than the value you answered for question 10? What did you put for your answer for question 11? Okay, good, folks, good. I think for the most part, the group of you are in the ballpark. Samantha, I'd be a little bit uncomfortable with 4 hours. You're getting at the upper range of things in there. So about 2.5 to 3 hours. Yeah, about 2.5 to 3 hours would be a good answer on that. Okay. Are there any problems? Anything we need to talk about so far? Are you okay? All right. Question 12. Which seems to be more inhibitory for enterotoxin B secretion, a basic or acidic pH? Hopefully you agree that it is basic pH. Okay, basic pH is more inhibitory. Just compare line D to line C. Compare line D to line C. And I mean, look at the secretion of line D there, right? It's at the floor. So, all right. Okay, and then we have everybody's fan favorite, and that's a joke, Elena. I know it's not a fan favorite. Question 13 there. What'd you put in for question 13? Okay, what did you guys put in? What did you gals put in for question 13? Okay. 1/3 of a day. 1/3 of a day. So obviously, you are all sophisticated enough to know that 8 hours is equivalent to 1/3 of a day. So you could have just reasoned that way in your mind, but could you actually do the unit conversion? Could you actually go through the unit conversion process to do that? Because that's what I was hoping you would do. Okay, so just to complete that here in our notes, let me do the unit conversion for you. And you know what? I think I'm going to do it on the PDF because my ability to draw on the PDF is just a little bit better than in Microsoft Word. Okay, So it would be like this, folks. Okay, for those that just reasoned it out in their mind, I get that. This was simple enough that you could do it. But if you had a more sophisticated problem, could you do the unit conversion process? So the idea is to take 8 hours and convert it to days. 8 hours times the conversion factor, right? So you're going to have 1 day is 24 hours, right? You can cross out the hours on the diagonal. That's going to leave you with 8 over 24, right? Which is 1/3 of a day. Okay. All right. So number 13 is 1/3 of a day. Question 14. All right. I'm curious how you reasoned with question 14. What do you say for question 14? How about the rest of you, gang? Okay. Anybody else? We got 3 answers. Can we get a couple more? Okay. Thank you for participating. I deeply appreciate it. And so let me say, go ahead and look at everybody's answer there. People are saying that fermentation is an anaerobic process. There's no oxygen, okay, so on and so forth. And those are very good answers for beginning biology students. But life is always funny, isn't it? Life can always come around and shock you. Here's the deal. Let me ask you this question in the chat window. Here we go. From information and workbook activity number 2, do you know the kind of cell the data refers to? Do you know the kind of cell the data refers to? Do you know the type of cell the data refers to? All right, you don't know. You don't know, right? There's no information provided here in this workbook activity. So let me ask you a question in the chat window. Here we go. If I told you that knowledge of the type of cell being measured matters for this question, would your answer represent an assumption on your part? Okay, so just humor me, folks, okay? What are the first 3 letters of the word assumption? You bet. Put it in bold-faced capital letters, right? Very good. So here's what I'm trying to say to you. Probably as beginning biology students, you've been taught that fermentation doesn't use oxygen, so that seems like a reasonable assumption to you. That's what we teach beginning biology students. But folks, it is not universally true about what we're saying here. What I'm telling you is that fermentation can take place in environments that still has oxygen, but the oxygen is simply not being used. For example, let me tell you in the chat window what kind of cells can ferment even when oxygen is present. Here we go. Cancer cells. Cancer cells can ferment even when oxygen is present. Okay, so cancer cells, and there's a whole treasure trove of various kinds of microorganisms on the face of the planet that even with an abundance of oxygen available in the atmosphere, will still simply not use the oxygen and perform fermentation anyway. Okay, so this is one of these, oh, I gotcha, okay? And it's just meant to show you that we all, myself included, we make assumptions about everything, right? We make assumptions that color our perspectives of the world. And so the honest answer for 14 is you can't really tell because you don't know the type of cell the data is referring to. Okay? You can't really tell because you don't have enough information. And part of science is appreciating when you know what you know and appreciating when you don't know what you don't know. And that's a hard thing for us humans. Okay. All right. So folks, question 15 in the chat window. What did you get for what did your team get for question 15? Okay, all righty then. All right, so just take a look across the chat window there. You can see that there is not agreement. There is most definitely not agreement in everything. And so let me say, well, Rachel, your answer is correct. Alicia, your answer is not correct. Taylor, your answer is correct. Chelsea, your answer is not correct. Elena, your answer is correct. Okay. Lance, your answer is correct. Victoria, your answer is correct. Odalis, you're good. Aubriana, you're good. Yasmin, that there's a problem with that. Okay. All right. So if you don't understand how to do this, please seek out some help. Please come see me. And for those of you that are knocking this activity down, good for you. Good for you. Okay. All right, folks, any questions before we continue? All right. Okay, so at this point, I'm going to stop the recording because I'm officially done with lecture. Okay, so we'll stop here.